MIB Agents OsteoBites webinar and podcast presents the latest in osteosarcoma treatment, research, innovation, and hope each week. Today, we're speaking with Dr. Alejandro Sweet-Cordero about genome-informed targeted therapies for osteosarcoma. Our panelists today are Junior Board member Charlotte Murdoff and Junior Board alumni Ryan Kennington. I'm your host, Anne Graham. So welcome to OsteoBites. I hope you have your snack. I have mine. It's chocolate covered almonds. Don't judge. And also a cappuccino because I got to keep up with Dr. Sweet-Cordero today. So I need all the help I can get. Today we are speaking with Dr. Alejandro Sweet-Cordero. He is of the University of California, San Francisco, Benioff Children's Hospital, and so much more, which I hope he'll fill you in on. We'll be talking about genome-informed targeted therapies for osteosarcoma. As usual, we are an all Osteo Warrior panel with MIB Agents Junior Board member Charlotte Murdoff and Junior Board member alumni Ryan Kennington, and I'm your host, Anne Graham. I'm also an Osteo Warrior and president of MIB Agents. MIB Agents is a leading pediatric osteosarcoma nonprofit dedicated to making it better for our community of patients, caregivers, doctors, researchers, all with the goal of less toxic, more effective treatments and a cure for this aggressive bone cancer. Our mission is in our name, MIB, make it better for kids with osteosarcoma. We make it better three ways. The first, through direct patient and family support, through education, through our annual Factor Conference, OsteoBites, and our book, which, by the way, now available in Spanish, as well as Chinese and English. If you need copies, just let us know. If you want to just download it, it's free on our website. Let's see. And then we also share and fund meaningful osteosarcoma specific research. If we can make it better for you, please let us know. If you would like to join our shared mission to make it better, we want to hear from you. Whatever it is you do it for kids with osteosarcoma. Whatever magic you have, we need it to improve osteosarcoma. With that, Dr. Sweet-Cordero, would you get us started by introducing yourself, please?
Sure, and first just to say thank you for the opportunity to be here today. This is fantastic. This is a wonderful group. It's always a pleasure to be in any Make It Better event. We were just talking about how one of the last in-person events we were at was in Phoenix, which was a lovely time that I remember very fondly. I think I specifically remember Dr. Hellman trying to eat a donut from a string. That was maybe the highlight for me.
Me too.
But
that was awesome.
Yeah, that was quite entertaining. So my name is Alejandro Sweet-Cordero. I'm a physician scientist here at UCSF. I was a medical student and a resident here, and as a resident here at UCSF, had an opportunity to start working with many patients with cancer, and really was motivated and became passionate about that, and so decided to go to Boston to do my fellowship, where I completed my fellowship in hematology oncology at the Dana-Farber Cancer Institute, and then came back to the Bay Area, where I was on faculty at Stanford for about a decade before moving back to my roots here at UCSF. And so I spent about 70% of my time doing research and the other 30% doing direct patient care. And I really enjoy both aspects of my job. I enjoy very much working closely with my patients and their families. As you said, I think making it better is really what it's all about. And then we do that by direct patient care and also do research, which is, a way to ultimately improve the treatments and the options that we have for all of our patients. So again, thank you for the invitation.
Hi, my name is Charlotte Murdoff. I'm a 3-time osteosarcoma fighter, currently undergoing cancer treatment for my 4th relapse. I was originally diagnosed in July of 2018, had limb salvage surgery above my right knee, and then all of my relapses have been in my lungs.
Hi, my name is Ryan. I'm 24 years old. I was originally diagnosed with osteosarcoma back in 2014. I had a limb salvaging surgery. I unfortunately had a relapse 1 year after that in 2015, but had VATS to remove only one nodule, and I've been luckily cancer-free ever since. And now I'm a full-time member of the Shiffman Lab.
So I realize this is a very diverse audience with diverse backgrounds in terms of expertise and knowledge of cancer biology. So I'm going to try to kind of go slowly here and explain the slides, make sure that we're all sort of on the same page. So the title of my talk today is Towards Genome-Informed Combination Therapies in Osteosarcoma. I wanted to just start by mentioning what I think are some of the key challenges that we face in this area. As I think most of you are aware, osteosarcoma is a highly heterogeneous disease, and what we mean by that is that no two tumors are alike. Obviously, no two patients are alike because every person is unique, but also every tumor is unique in its own way. Also, fortunately, overall, osteosarcoma is a rare disease, which is a good thing, but also makes it difficult to access tissue and samples to do the kind of research that we would like to be able to do to move things forward faster. We also have a lack of very well studied or well described preclinical models. So for some diseases, for example, lung cancer, pancreatic cancer, common cancers like that, there are lots of preclinical models, either cell lines growing in plastic or mouse models, or sometimes even zebrafish models that can be used. But we don't have that depth of preclinical models in osteosarcoma, which is another barrier. And as many of you are also aware, access to drugs is difficult, and particularly when these are investigational drugs, requires intensive negotiation with drug companies who oftentimes don't have a huge incentive to push forward clinical studies in osteosarcoma because they're focused on the more common diseases. Lastly, I think a very important point is that although we've learned a lot about osteosarcoma, the number of researchers working in this field is still relatively small, and therefore there's a lot about the biology of this disease that we don't really understand very well. There's lots of examples of that. So what makes it so heterogeneous? Why is it so genomically complex? Why does it have a propensity to go to the lung? What are the determinants of that propensity? These are all things that we don't understand very well. So in my lab here at UCSF, well, first at Stanford and now here at UCSF, thinking about some of these barriers, we've developed a program that we've had now for about 7 years, where we're trying to start out by basically collecting as many samples as we can, with really the goal of trying to understand sort of the landscape of the heterogeneity of this disease, both in the primary tumor, which is kind of highlighted here as a tumor in the femur, and then the metastasis, which oftentimes are in the lung. And then we take those primary samples and we do a couple of things. One is using technologies that allow for sequencing of entire genomes, which have become now frequently used and many are familiar to many of you. We can sequence both the whole genome of the tumor as well as what's called the transcriptome, which is the part of the genome that is turned into RNA. And so we can do both the DNA sequencing and the RNA sequencing, and these are just some figures that kind of highlight some of the data that we can see when we do those kinds of experiments. We can also then take these tumors and implant them into mice, and those are called PDXs, which stands for patient-derived xenograft. And the reason we do that is because, not because we like to torture mice, but because, as I said, it's a rare disease, and therefore there's not a lot of tissue available. So we basically utilize the mice as kind of a carrier to expand the amount of tissue we have available, so that then we can do experiments that obviously we wouldn't be able to do in the patients themselves. So once the tumors grow in mice, we can do the same experiments, the same sequencing that we do in the primary human tissue, the whole genome sequencing, the RNA sequencing. More recently, and I'm going to show you some data today, we've started to generate cell lines. And what a cell line means is when you take this tumor out of a mouse, you can put it into what's shown here as a little plastic dish. This red stuff is special media that is basically just dyed red so that you can see that it's there, so it's not clear. And also when it's when the cells have kind of secreted a lot of toxins, this red dye turns yellow, so that's how you know you need to change the media. So that's why you see this color red oftentimes. So this basically on the right side gives us what we call models. So these are models of the disease that we can then expand, and if we want to say try a certain drug, either in the PDX directly or in the cell line, we can do that. We've also done a lot of work, which I'm not going to have time to talk about today, to try to understand, if these tumors are implanted into mice, do they have a tendency to metastasize within the mouse? And so can we use that metastatic capacity in the mouse as something we can also test drugs against? And so we're doing a lot of that work as well. So this is a little bit of a complicated figure. It's this little wheel that kind of summarizes all of the work that we've done over the last few years that I just described. This is updated to December of 2020. So each one of these little slices is a particular PDX model that we've developed. And the way to read it is if you start from the outside, you can see whether these are multiple samples from the same patient. And you can see whether we have this blue circle indicates that we have blood stored from that same patient. The yellow indicates whether that patient was metastatic at diagnosis or became metastatic later. And then we can also indicate where the sample came from, and then what kind of sequencing has been done, what kind of analysis has been done. So we just kind of use this to keep track. And the reason we're, again, trying to develop lots of these models, you can say, well, why do you need so many? Why can't you just use one or two? Is again, because of this heterogeneity that I was just talking to you about. So we've been doing this for quite some time, and now we've developed over 20 models that we've characterized quite deeply, that we know we can passage from one mouse to another, and we're continuously trying to develop more. And these are resources, I should say, importantly, I think one of the really important things about this research community that works on osteosarcoma is that we really try to share these resources. I don't consider these to be my resources. These are obviously, tumors that came from patients who are suffering from this disease. So it's not really my property to keep. So if anybody asks me for them, I'm happy to share them. So we share them with lots of labs. We've probably shared with over 10 labs, and we just got another request last week. So if anybody has a particular question they want to ask in terms of research and wants to work with these models, I should say that we're not the only ones that have made these kinds of PDX models. The group at St. Jude's, the group at Sloan Kettering, the group at Baylor, I think are all developing these models. So I think together we can, we now have a pretty large collection that I think can be used to ask a variety of very important questions that will make it better and that's the ultimate goal. So here's where it gets a little bit deeper into the science, so I apologize for that, but I'm trying to walk you through this. So one of the things we know about osteosarcoma is that it's well, you've probably heard this term thrown around a lot, genomically complex disease. And so what we mean by that is that the genome of osteosarcoma has a lot of abnormalities. And so one of the key abnormalities we know is very common is what we call a copy number change. So this graph here indicates each one of these colors is a different chromosome. So the human genome has 22 pairs of chromosomes plus the X and Y chromosome. So each one is shown here in a different color. And in a normal cell, like if you took cells from your blood and you did whole genome sequencing, you would basically just see a straight line across here because every cell in your body is supposed to have two chromosomes, one that came from your mom and one that came from your dad. But here in these osteosarcoma genomes, you can very clearly see, you don't have to be a cancer biologist to see that this is not a normal pattern. And so you see lots of these kind of jumps up and drops down. Those are called chromosome arm amplifications or chromosome arm deletions. And then you also see these kind of very steep peaks here. Here's one, here's another one, here's another one. And those are called focal amplifications. And so our work and the work of many other labs has shown that these focal amplifications probably are key driver, what we call driver events. So they contain genes that the tumor has decided, I really like this gene because it really helps me grow faster, it helps me do the nasty things that I want to do. And so I'm going to get, instead of having 2 copies like a normal cell, I'm going to make, in this case, 40 copies or 50 copies. And so a lot of our work is really directed at understanding these amplifications, what genes are in them, what can we do about them, how can we target them. But importantly, what I'm trying to show you here is this is the metastasis from the patient. This is the PDX from the same patient in the first passage. And then we took it out of a mouse and put it into another mouse, made the second passage. And then from that, we can make a cell line. And if you look across down this, you'll see that, for example, this very steep amplification of chromosome 8 is present in the metastasis. It's also present in the PDX. It's maintained in the PDX, and it's also present in the cell line. Here's another one on chromosome 11, same thing. It's not always the case. So for example, here in chromosome 1, you see an amplification that wasn't detectable in the metastasis, but now it is detectable in the PDX. And whether that's because we selected a small sample of the patient sample that then grew out and gave rise to this, so you would have actually seen it in the patient if you had found the right fragment, or whether this is something new that wasn't present in the metastasis but is occurring in the PDX. So it's really hard to answer, but overall, in general, we find that these cell line models and PDX models very closely reproduce what we see in the patient. So that we take to be a good thing, and that what we learn from the cell lines will potentially be helpful to the patients. Another way to look at that, so this is what I told you before, is copy number data. So this is the DNA, right? We can also look at the level of RNA. So these are the genes that are turned into RNA, and RNA is all the rage now because that's how the COVID vaccines were made, from RNA. And if you remember from your biology classes, DNA makes RNA, and RNA makes protein. And so that's kind of the central dogma of biology, so-called. And so what RNAs are made in a cell is actually very important. So here, what we use this technique that I was telling you about earlier called RNA sequencing. So instead of sequencing the DNA, we're only sequencing the fragments of the DNA that are being transcribed into RNA. And this is something called a dendrogram. And again, I'm trying to get into too many details, but basically it's a way to show similarity between 2 samples. So the closer 2 samples are in this dendrogram, the more similar they are. And the further away they are in the dendrogram, the less similar they are. So for example, if you look at this sample 457, it's very similar to another sample here 457 and this one 457, but it's very different from this sample over here 152, because you have to go all the way up the dendrogram, all the way up here, all the way here. So the further the distance you have to travel horizontally and then vertically and then horizontally again, the less similar 2 samples are from each other. So basically you can see that's what we label here 1, 2, and 3. We see 3 main clusters. We call them clusters because the tree starts at the left and then it kind of breaks into these branches. So the top 3 branches are branch number 1, branch number 2, and branch number 3. And so what was really exciting to us when we did this is that if you notice here, you'll see that the patient cell line, the patient PDX, and the patient sample itself, 457 is one patient, same patient. 357 is the same patient. 52 is the same patient. 384. So the patient and the PDX, and when we have a cell line, they tend to cluster together, suggesting again that the cell line and the PDX is a good model for what's happening in that patient because the RNA is closely correlated. In contrast, cell lines that are often used and have been used by many labs for many, many years, including U2OS, SJSA, MG63.3, which are what we call commercial cell lines. You can go to a website and you can buy them and have them shipped to your lab. and they were made from patients maybe 30 or 40 years ago, they've kind of evolved away and they don't really look like the patients anymore. And so this is another reason why we're sort of confident that what we're doing in these patient-derived cell lines is hopefully relevant to the patients. And this is just kind of a zoom in of the same thing so you can see it a little bit better. So we've made this panel of cell lines that are shown here. And again, I just wanted to take a moment to thank all of the patients and the families who contributed this data or contributed these samples and were willing to sign the consent so that we could take the residual tumor removed from them at surgery and use it for this research. So just wanted to thank the people, anonymous obviously, but who contributed to this. And so we've been able to then analyze these samples using the sequencing technologies that I described. So we know what the mutations or amplifications or those high peaks that I showed you on that prior slide. We also know where they came from, both the source, whether it was a diagnostic sample or metastatic sample, the specific location, the age of the patient, the sex, and the doubling time, which is basically just how fast it grows in tissue culture, which is important for us for technical reasons. And just again, to kind of highlight the complexity of osteosarcoma as a disease, this is from a panel from a figure of a paper we published a few years ago. Again, kind of complicated to describe, but basically each one of these columns you see here is a specific sample taken from a specific patient. Some of them are from our lab that we sequence them. The ones that say SJOS are from St. Jude's. And again, this just highlights the importance of data sharing to be able to learn things about these diseases. And so here, as I said, every patient is a column and every gene is a row. And when something is dark red, it means that it's highly amplified, meaning it's got one of these high peaks that I showed you before on this slide, so one of these kind of things. And if it's not dark red, if it's just yellow, then it's sort of a lower level amplification. And then these blue things are deletions, so these are genes that are being lost as opposed to gained. And then there's just a lot of characterization here. So again, this just highlights the heterogeneity. So no two patients are identical. Every patient kind of is different from every other patient. But at the same time, you also notice, for example, genes like MYC, genes like VEGF-A, genes like CDK4 are frequently amplified in many patients. So even though there's a huge heterogeneity and every patient is unique, there are commonalities across patients. And so that's what we're trying to exploit. We're trying to find those commonalities. So rather than think of every patient as its own disease, can we put patients into groups, or what we sometimes call buckets or baskets, so that we say, okay, well, these are the CDK4 amplified samples, these are the MYC amplified samples. And so maybe we can find drugs that work for those subgroups of patients. Here's just another way of looking at the same data. So rather than looking at it patient by patient, you're just looking at the frequencies. And again, what's important to point out is that it's not just kind of like a random madness in genomes, that there's random amplifications. There are specific regions in the genome, this is again, all the chromosomes, that are frequently gained or frequently deleted. And so when we see those kinds of peaks and valleys, that kind of tells us, okay, that's something that the tumor really wants, and there's got to be a reason for it, right? It's not just random. If it was just random, then we would just see, kind of everything blue and everything yellow. So the fact that we see these very high peaks and valleys kind of gives us a strategy to move forward to try to find genome-informed therapies. So this, again, work that we published a while ago, and I apologize, many of you have probably seen this, but I wasn't sure how many of you haven't. So again, we then can put patients into groups with, depending on what key amplified genetic event we see, we can match that to drugs. We can then, these are the pathways that we think are important that we're trying to inhibit. And so here's, for example, two patients that have very high, two PDXs that have very high levels of MYC amplification. So this is just rank ordering the various genes that we think are important. MYC is at the top. This is something called a Western blot, which is at the protein level. So remember, I already talked to you about DNA, I already talked about RNA. We also look at protein. Unfortunately, protein is harder to look at kind of at large scale. There's no way to like sequence protein per se. Well, there is, but it's difficult to do. So we tend to just look at one protein at a time. But here you can see, again, you don't need to be a cancer biologist to see that there's a very strong signal here. This black line, black blotch here indicates that there's lots of MYC protein in these two samples, but not very much in these two samples. So that to us suggests that these two tumors, for whatever reason, really like to have lots of this protein called MYC, which was something we know is a so-called oncogene, which is a protein that is very well known, very well described in thousands of publications to be an important gene for driving cancer. So the fact that it's expressed here and not here suggests that, well, maybe if we could find a way to block it in these two patients, we could help control their disease. So of course, these are not patients, these are mice. But in these two mice that have high levels of MYC, we were able to control their disease pretty well, as you can see here with this green line, with this chemical. I'm not going to call it a drug because it's, for a variety of reasons, probably not ever going to make it into a patient, but it's a chemical that we can put into mice. And so it's kind of proof of principle that this works. And here's another kind of more experiments kind of showing the same thing. I'll skip that for lack of time. Here's another one where CDK4 is highly amplified. You can see lots of protein there. And same thing, is kind of a flat line in this PDX, maybe not so much here, but still an effect. So this kind of summarizes all the data that we've already published. So now where do we go from here? How do we take this to the next step? These are all tumors and growing in mice that we treated with a single drug. Obviously, I think everybody in the audience probably knows that we feel strongly, and I think most people feel strongly, that we're not going to cure osteosarcoma with one drug. So how do we start moving this towards combination therapy? So that's really what we're doing in the lab now. As I said, single agent chemotherapy is unlikely to cure osteosarcoma. Because osteosarcoma is highly heterogeneous, we need to kind of base our combination therapies on some of these so-called biomarkers, which are these oncogenes that I was just telling you about. And the frequently used cell lines, which are the ones I told you were sort of commercially available, may not fully reflect this heterogeneity, and I think some of our data supports that. So, but of course, we also know that even if we find some combination therapies in cell lines or mice, transitioning that to an actual clinical trial is a heavy lift and something that we obviously need to work on. But so what we started doing was taking this panel of cell lines that I showed you we made from these PDXs and just expanding the number of proteins that we look at in these so-called Western blots. Again, here's MYC, high in some tumors, low in others. CDK4, high in some tumors, low in others. And a lot of other proteins, genes, again, oncogenes that we think are relevant to the biology of this disease. Again, they all show heterogeneity. So here's another one that's familiar to many of you, RB, absent in a couple of PDX cell lines, present in others. And so, here's where things start to get kind of complicated, right? So how do we, now you can imagine, well, every patient is kind of got its own pattern, right? No two patients are identical, even at the level of the protein. So how can we start to put those into groups that we might be able to decide what's the best combination therapy? So that's kind of what we're doing as I speak. So, this is ongoing research. All of this work that I'm telling you about here is unpublished, ongoing work in the lab. So this is now taking that panel of cell lines and treating them with a much larger panel of drugs that we came up with in consultation with other experts in osteosarcoma, particularly Dr. Katie Janeway, who's been a close collaborator of ours. And again, this is a complicated figure, which I won't spend too much time on, but just to say that what we're looking for is small pink dots. So when you see a small pink dot, that means the drug is highly effective in that cell line. So you see some drugs that seem to be effective across all the cell lines, for example, this one here, but then there's other ones that are highly effective in some cell lines, but not in others. And again, this is all single agent therapy. This is data that I've shown before. I think I actually showed this at the MIB meeting. And so now we move to, kind of the real meat of this, which is the combination therapies. And this is, again, a very complicated slide that I'm not going to spend a ton of time on. It's pretty new, recent data, but this is the result of combination drug screen, over 700 drug combinations, 10 cell lines, 14 targeted agents, combined with the backbones, so-called backbones that you show here on the right. So we took all of these targeted agents and combined them with each one of these backbones. And then we use this is all work done in cell lines, in vitro, using kind of high throughput robots that help us to do all these 700 drug combinations and multiple replicates, and then kind of summarize the data in the way I'm showing you here. So basically what we're looking for is things that are what we call synergistic. So if something is synergistic, it means that at a certain dose, drug A works, and a certain dose drug B works, but when you put them both together, they work as well, but at a lower dose. So that's what's called synergy. And if something is antagonistic, it means that if you give them individually, they work, but if you put them together, they work not as well. So that's obviously something we want to try to avoid. So we're trying to avoid the red things, and we're trying to pick out the blue things. So if something is deep blue, it means it's highly synergistic, and if it's deep red, it means it's highly antagonistic. So one thing you'll notice, for example, is that gemcitabine seems to be highly synergistic with a lot of different drugs. So that's kind of interesting, because gemcitabine is obviously a drug we use not infrequently in osteosarcoma, and it's a drug that is easy to give and easy to obtain. So we don't need to negotiate with drug companies in terms of getting gemcitabine. So that was part of the reason for using these as backbones, was the idea that, well, we don't want to have to negotiate with two different companies to go to the next level. We want to be able to just negotiate with one company. And so we also tried to cover as much space here in terms of different mechanism of action. So the various genes that are inhibited by these drugs are shown on the left side here. So again, this is very new data. We're doing now a lot of validation to follow up on these drugs and to see which ones kind of seem to work the best. And the goal now is really to take them to further testing in the mouse models. Here's just one example of one cell line we labeled as 08186. And you can see that with these various backbones, the combinations that are highlighted in the blue boxes, basically the single agent gives you either the red, the black, or the gray line. So we're basically looking for things that shift the line towards the left. So that's indicative of synergy. So for example, here, gemcitabine, very clearly you can see that with this drug, we see a shift to the left, whereas with this drug, we don't. So those are the ones we're most interested in, are the ones that synergize to the left. So here's this one combination, gemcitabine plus a CHEK1 inhibitor, again in the same cell line, 186. Again, these are more Western blots, trying to get more at sort of understanding the mechanism. One of the things we're really looking for is something called cleaved PARP, which is a protein that is only expressed in cells when the cells are dying. And so obviously we want to kill these cells. So if we see lots of cleaved PARP, we get all excited and jump up and down, because that's telling us that this combination is not only stopping the growth of the cells, it's actually killing the cells. And so that's actually something that we very much like to see. Here's just kind of an example of a one xenograft experiment that we have done now in a tumor in a mouse model of this particular combination, palbociclib, which is a drug some of you are familiar with, which is a CDK4 inhibitor, and irinotecan. Kind of encouragingly, we see that when we combine those two drugs, again, we see kind of a flat line of the tumor volume. So the tumor kind of stops growing. When we stop the drug treatment, then the tumor starts to regrow. So we would, if we wanted to say cure this mouse like we would want to cure a patient, obviously we would not just give them one cycle. We would do what we do in patients, which is give them multiple cycles. But here is just a proof of principle that we do indeed see a decrease in growth when we do the combination. So what are our current efforts? So as I said, we're doing the work to extend the validation to what we call a full matrix of drug doses to define the synergy, and then move those forward into further work in PDX models, either primary or metastatic. We're particularly interested in much of the work that we've done so far, I should say, has been done looking at growth of a primary tumor in these mouse xenografts. But I think you would all agree that what we really need to cure in osteosarcoma is not the primary tumor, because we know how to cure that. The combination of surgery usually takes care of it, surgery and chemotherapy. What we really need to cure are the metastatic tumors. So we're actually developing these metastatic tumors in the mice so that we can actually determine whether these drug combinations also inhibit growth in the lung or potentially in the bone, which is the other common site of metastasis. And then obviously trying to determine the biomarkers. So now that we know which cell lines respond to what combinations, we can go back and look at the sequencing data to try to infer what is the likely genomic driver of that combinatorial response. So that's obviously work that we're still doing. So just to conclude, I think everybody here will agree that osteosarcoma is characterized by a complex genome. But our work and the work of many other labs suggests that this complex genome, although it's in some ways a detriment, it also may provide an opportunity if we can really understand what are the key drivers, oncogenic drivers for each patient, and that this kind of idea that we've helped to develop of a genome-informed targeted approach may be a path forward for this disease, and that developing these PDF models is kind of a way to test that hypothesis, and we've identified some specific combinations that we're particularly excited about. So I just wanted to end by thanking all the people in the lab who have done all of this work, particularly the people shown in red who are kind of our osteosarcoma team, Leanne Sales, Courtney Schott, Betsy Young, I should forgot to put her in red, Marta, Marcus, Alex, and Crystal, all of whom have done tremendous work and contributed to this. We also have a fantastic group of collaborators. One of the things that's most fun about science is we get to collaborate and talk to a lot of other people with exciting ideas. So we worked a lot with people at Stanford, University of Minnesota, other colleagues here at UCSF, Dana-Farber that I mentioned, MD Anderson and others. And then of course, our many external funding agencies, including the National Cancer Institute, St. Baldrick's Foundation, the Osteosarcoma Institute, Hyundai Hope on Wheels, Alex's Lemonade Stand, and many others who have supported our work over the years. So with that, I'll end, and I'm happy to take any questions.
Okay, so I had a question. You said that you test certain drugs or chemicals in mice that wouldn't be used in patients. Why would you test those if they can't be used in like real patients?
Great question. So there's a variety of there's a kind of what we call a pipeline for development of a drug from, say, discovery to an actual drug that goes into patients. And so oftentimes there are drugs that are kind of in different or chemicals that are in different stages of that pipeline of development. So maybe they haven't been used in patients because though the drug is still in a phase 1 trial, or maybe it hasn't even gotten to a phase 1 trial, it's still being developed by the drug company, and it's being developed because it has an important mechanism of action that we think might be important, but it hasn't made it sort of all the way through all the safety studies that need to be done. And sometimes what happens is a drug looks really promising early on. This is kind of what happened with AT7519. It looks promising early on, but then when they start doing clinical trials, phase 1 trials in patients, they realize that it has, for example, a significant toxicity, right? So that kind of kills what we call the clinical program for development of that drug, but it's still a useful compound in terms of understanding the biology of that target. And maybe if we kind of push that, we can convince the company to, well, pick it up again and maybe change the formulation so that it doesn't have the toxicity anymore, but it's more effective. So there's kind of, that's the rationale, but that's a great question.
Do you believe all osteosarcoma patients should try to get their tumor genes tested? And would this provide benefit to a patient who has had several relapses?
Yeah, great question. So 2 questions. Does every patient with osteosarcoma, should they get their tumor sequenced? This may not be a universally accepted idea, but I think it's more and more accepted. I feel strongly that the answer is yes, that every patient, even at diagnosis, even if they have non-metastatic disease, should get sequenced. And there's a couple of reasons for that. One, in osteosarcoma particularly, the incidence of what we call a germline predisposition is quite high. In our cohort of patients that we've looked at here at UCSF, it's close to 30% of patients with osteosarcoma have a germline predisposition. So what we mean by that is it's something in your normal genome, not in the tumor, that predisposes you to get cancer, right? So the most common, well-known example of that is something called Li-Fraumeni syndrome, which is a mutation in a gene called p53, which is well known to predispose you to osteosarcoma. But importantly, it also predisposes you to other cancers, right? So it's important for you and also for potentially other members of your family to know that you have one of these predispositions, and also to know that you don't have it, right? So you can maybe not have to worry about that. So I think that's one very important reason to test everybody at diagnosis. I think in the relapse setting, it's even more important because I think more and more there are examples of how you can use that information to help guide therapy. So, for example, in the case of CDK4 amplified osteosarcomas, there's now several case reports that show that those patients can respond to CDK4 directed therapy, and there could potentially be other things. So I think and then I think thirdly, I think of course this is a philanthropic question, maybe not particular to the patient, but I think because this is a rare disease, it is very important that we get more information so that we can understand, at least what we call retrospectively, are there certain markers that are making more likely or less likely to respond to a certain therapy. So I think there is a very good reason to do the sequencing. The other question that comes up is what kind of sequencing? I think that was the second part of the question. Do you do whole genome sequencing, this kind of more complex research-based sequencing that I'm talking about here? That is a more difficult question. I think most people will get sequencing that is what we call, kind of DNA panel testing or some kind of commercial vendor, like something like Foundation Medicine. Here at UCSF, we use a test called UCSF 500. Sloan Kettering has its own test called the Impact test. I think other institutions have their own test. So I think some kind of a panel assay is, I would say, very strongly recommended. Participating in research studies that are doing whole genome RNA sequencing, I think, is useful. And occasionally we do find it to be helpful for individual patients, but I don't think we've done enough research and with enough patients to be able to sort of categorically say, yes, everybody should get whole genome sequencing, or yes, everybody should get RNA sequencing. I tend to be biased, and I think that it can be helpful, but I think it's not been sort of definitively proven.
Are many patients' primary tumors sequenced, like even before the MAP chemo is given? Because it sounded like most sequencing is done after the cancer has metastasized.
Probably still more common, when obviously when patients have metastasis, they're searching for other options. And so there's kind of a more, I guess, incentive from the part of the patient and families, and maybe also the physicians to do the sequencing. So I would say that not everybody is getting sequenced at diagnosis, but I personally think that it is helpful to do it early on.
Is there any difference in the METS models by site, like lungs versus bone?
We do see that in the models, in the mouse models, every cell line behaves a little bit differently. So some cell lines, for example, will metastasize to the lungs. Some cell lines don't metastasize at all. Some metastasize to bone. And so we're still trying to understand that. And I think that's actually something that's quite interesting from a research point of view, but how relevant that is to patients, we don't really know yet.
Katie Moreno asked about insurance covering the sequencing for patients. And I can answer that quite simply. If you go to the one that I know is Foundation Medicine, they have a spectacular financial aid program. I would go to Foundation Medicine for that.
Yeah, I agree with that. Although I will say that a lot of institutions, including ours, we tend to favor our own tests for a variety of reasons. And we also work closely with insurance companies to make sure that it's covered. So it's not only Foundation Medicine, but in a lot of smaller, particularly smaller institutions that don't have their own research efforts, Foundation is a great option. And so I agree with them.
How far away are we from having those sort of baskets for treatment? So if you have the testing, you're MYC amplified, you're VEGF-A, how far away are we from quantifying that at the outset and going, okay, well, listen, doxorubicin is not going to work for you. So we're going to do gemcitabine with cisplatin or what have you. How far away are we from that? Part one and part two. How can we expedite that? How can we make that happen faster? Because that seems like, from one standpoint, from the patient standpoint, if I don't have to have doxorubicin, I'm just throwing that out there. That's not the case. If I don't have to have a certain component of chemotherapy that's going to cause long-term side effects, going to make me sick in the meantime, be really great to avoid having to have that one component out of the four.
In general, I'm an optimistic person, but I think that's going to take a while. I think what's more likely to happen early on, sooner rather than later, is in the relapse setting, being able to define subgroups that are more likely to benefit from a certain combination versus another. Whether that's going to happen in the upfront setting, I think that's going to take a long time, mainly because MAP chemotherapy actually is pretty effective, right? Particularly in the non-metastatic setting. So convincing clinicians and to change that is going to be difficult, right? And we've tried to do things like as many of you are probably aware, there was a very large study that was done in collaboration between Europe and the United States to try to stratify patients based on their percent necrosis at resection, and the patients who had poor resection got i.e. ifosfamide etoposide early on, and the ones that had good necrosis didn't get it, and that turned out to make no difference, right? So that there wasn't like they did better when they got one or the other. So I think there's and that was a very large study. It took many years to do, and it was across two continents. So I think it's going to be difficult to move it into the frontline setting. I think it'll happen eventually, but if you wanted me to give you years, I'm thinking probably 3 to 5 years will be my guess. I think the defining early options for metastatic patients is probably going to happen sooner. Maybe that's more in the kind of 2 to 3 year range.
Someone asked, we have seen a gene splice shown up in Foundation 1 testing. What is a splice compared to like an amplification or loss?
Oh yeah, that's a great question. It's a little bit complicated, but to try to answer the question in an easy way it's easy to understand. In your DNA, we talked about genes, right? So genes are encoded in DNA, but they're not encoded in a kind of linear way. So let's say that you all know you have 4 letters in your DNA, and it's a combination of the 4 letters that determine which gene they correspond to. But it kind of comes in chunks. So there'll be like sort of chapter 1, chapter 2. Let's say there's 4 chapters, chapter 1, 2, 3, and 4 in this particular gene. And in the middle, there's kind of like empty pages. There's like, because that's just the way the genome works, there's 20 empty pages. So there'll be letters and 20 empty pages and more letters and more empty pages. And so those empty pages in between the chapters are called introns, and the chapters themselves are called exons. So, and when a gene gets turned from DNA into RNA, the exons get spliced together and the introns are removed. So now you end up with what's a real book that doesn't have empty pages. You have chapter 1, chapter 2, chapter 3, and chapter 4. Sometimes a gene will have 2 versions. There'll be one version that is chapter 1, 2, and 3, and there's another version that's chapter 1, 2, and 4, and that's called a splice variant. So there's splice variants. So in one case, the chapters get spliced one way, and another way they get spliced a different way. So there are some mutations, and these are called splice mutations, that drive that splicing in one direction or another. So if you have a splice variant, you might never see chapter 4, you only see chapter 1, 2, and 3.
As I understand, you have derived cell lines from both primary tumor and metastatic sites. And so with respect to the drug responsiveness done to date, what is the general agreement across these differentially derived cell lines, and have you identified any unique responsiveness with metastatic lines?
The bottom line is, the honest answer is we don't have enough lines of any one group to really be able to say for sure. So we only have 9 lines total. I think we have three metastasis, 3 diagnostic, and 3 resections. So I think that's a great question and something we're very interested in asking. But I think we probably need more cell lines to know for sure whether we have a drug that works really well for metastasis or really well for primary tumors. But that's definitely something we're interested in looking into.
Quick one on how to determine driver genes of osteosarcoma. Are the genes the result of natural selection or of the tumor cells?
Yeah, I'm not sure natural selection is the right term, but they are definitely selected for during the evolution of the tumor, which is why we see them recurrently, right? So, as I said in the talk, we don't see sort of random gains and losses in the genome. We see that things like MYC, CDK4, VEGF-A, those are very commonly amplified, right? So, it's a complicated process, and I don't think we totally understand it. But there are mechanisms that sort of drive these cells as they're dividing to sort of accumulate specific alterations, such as MYC, for example, right? So it is a kind of there's sort of selective, what we call selective pressure for gaining those things.
Another quick question: is this data collected and shared across different countries, Austria specifically, but China? Like, how is it
yeah, great question. So the data that we've published is freely available through a repository of data called EGA, which is actually in Europe somewhere. So, but that's kind of data that is what we call raw data, right? So you kind of have to know what you're doing to work with that data. It's not something that a typical person without a lot of computational expertise would know what to do with. But we actually had a grant from the Alex's Lemonade Stand that we just wrapped up last year, which was specifically geared towards making this kind of copy number data available and organized and shared in a way that was more easily accessible. And so we're in the process of kind of wrapping up that sharing website portal. So that's something that is going to be more widely available to people without a lot of computational expertise. You still have to kind of know what it's not sort of, you have to kind of have a little bit of background, but less so. But there are also other data sharing platforms. St. Jude's has a very large one called Pecan that also contains a lot of data. There's another one called R2, which is hosted in the Netherlands. So there are many efforts to do that in a variety of ways.
This approach makes a ton of sense, but it seems that a lot of trials today still report on results without including insight on the genomic profile of patients. Is there any insight on if this is changing, and can the COG provide guidelines? Because it seems like
yeah, that's a great question. And I think many of us are very much in favor of that approach. So the idea that all clinical trials should include a genomic component, right? So that we can learn how to use this information better. And I think that is more frequently being done now. I don't know that it's universal, but I think there is a strong push in the COG to do that. So, for example, we have a trial, which is not a COG trial, but we have a trial that we're running with Dr. Janeway at Dana-Farber and also here at UCSF that uses a couple of targeted agents. One is a PARP inhibitor and one is an inhibitor against something called ATR. And that trial does have this this genomic biomarker component. So the idea is that, we're not going to use it prospectively because there is some concern about, since we don't know enough about the biology, that if we use something prospectively and we picked, say, well, we're only going to give you this drug if you have the MYC, or we're only going to give you this drug if you have CDK4, that we might be missing some opportunities and some responses that we don't really understand yet. So I think there's more interest in doing it sort of retrospectively rather than prospectively. So go ahead and treat everybody the same for now, but collect the information so that retrospectively you can go back and say, oh, well, these 5 patients out of 50 that we treated had just an incredibly good response. Can we go back to their genomic data to try to understand why they had such a good response? And then in the future, we can incorporate that prospectively.
Okay, cool. All right, Ryan has 2 quick burning questions for you, and then Charlotte will have 2 quick burning questions, and we'll wrap it up.
Okay. What was your first job?
My first job was well, most of you young people don't even know what this is, but there used to be something called a paper boy who used to go around on a bicycle throwing papers into, and so that was my job.
Wait, paper? What is that?
Yeah.
What is that device?
And the last one, what do you hope for?
What do I hope for? Better lives for patients with cancer and better treatments and more cures.
Okay. Would you rather repeat high school or med school?
Med school for sure. I love med school. It's a lot of fun.
And
I don't want to go back to high school, nothing.
And what other profession would you like to attempt?
I want to be a flamenco guitarist.
Ooh, better get practicing.
That's what I really want to do. This is just my side gig.
Do you play guitar?
I try.
Okay, so that's our entertainment at our next Factor conference. Okay, with that, we'll wrap it up by saying that for the 5th year in a row, MIB Agents is awarding $100,000 for osteosarcoma specific research. As we've heard from Dr. Sweet-Cordero today, this kind of funding is essential to moving the field forward. So we are able to do this with thanks to our patient families through their MIB family funds. Those family fund members and our scientific advisory board will meet virtually this year, shortly, and vote on our top 3 finalists from all the proposals that we receive. Those 3 finalists this year, so exciting. We will be presenting those proposals to an OsteoBites audience who will then determine the winning project from the scientific advisory team and the family funds top 3. Truly an all osteosarcoma community effort to make it better. To submit a proposal, go to mibagents.org/outsmarting osteosarcoma. Next week, we're talking with Dr. Pete Anderson. He'll be going over the basics with us, thankfully, on how to read and understand labs and even radiology reports, along with general Q&A for osteosarcoma. And that's it. Thanks for joining us today. And of course, thanks to our guest, Dr. Sweet-Cordero. Amazing work, just brilliant research that I know we're going to rely on this. I know we rely on it today, and we're going to continue to rely on it even more in the future. Thanks to our panelists, Charlotte and Ryan, and thank you all for being here today. We'll see you next week. Thanks, everybody.
Thank you.
Next week, OsteoBites welcomes Dr. Pete Anderson of Cleveland Clinic. He'll be talking with us about understanding lab, path, and scan reports and other osteosarcoma questions answered. Be sure and subscribe to our YouTube channel where you can view our library of this and all OsteoBites topics and rockstar speakers. You can also listen to OsteoBites via podcast wherever you get your podcast.